So now we're going to talk about the terms homozygotic and heterozygotic and locus or loci in the plural term, and what those mean for chromosomes. So what I've drawn here are two separate chromosomes. These are homologous chromosomes for chromosome number 9. Now chromosome number 9 is noteworthy because this is what codes, this is where the allele is that codes for blood type. Okay, so we have two alleles for blood type, and the combination of those alleles gives us our blood type, A, B, O, or AB. Okay, so now we have how many of these chromosomes in a human cell? Well, we have 46, right? And I've drawn two of those. So do our chromosomes really look like these rods? Well, when you look at them with a microscope in their condensed form, well, yes, they do. But what do they do? What do they look like if you were to drill down? Use a microscope and to look down at this and see really what it looked like. It really won't look like rods like this. It would actually look like a sequence of DNA. Okay, so we can't draw all the DNA, wrapped around histones and everything else. We can't draw that level of specificity. So what we're going to do is we are going to show those as this rod-like structure we've shown here in the red and the blue. But in actuality, that is representing a very long stretch of DNA. Okay, so what do we know about DNA? Well, we know it has a sequence of bases, right? A, T, G, and C. So I've tried to, approximate DNA with the helical structure right here. Again, this is DNA. Okay. So what I'm going to do now is I'm going to show the sequence of one of these strands. So now we see the sequence of the top strand, and this shows us that it's what? AT, GG, so on and so forth. Now, does it make a lot of sense for me to draw all of these bases out? In actuality, it would go for thousands of bases in many cases. Would that make a lot of sense? Probably not. So what we do is we give these a specific letter. For example, we might give this one an A. Yeah, I'll call this A, an A allele. Now let's do the same thing on the bottom strand from the father. Now, what do we notice here? We see that it has the same sequence, right? AT, GG, so on and so forth. I'm going to call that one also an A allele. All right? So now we have two A alleles. Okay? A from the mother, A from the father. This individual would be type A blood. Because these sequences are the same. Do you see how these sequences are the same? We call this homozygotic. Okay? Homozygotic. It has the same sequence. They have the same allele at the same locus, which is the position on the chromosome. Okay? That's homozygotic. The same. Right? So same is homozygotic. Okay, so if this represented someone with type A blood and they had two A alleles, we would call them homozygotic. Now, if these two A alleles were dominant alleles, we would call them homozygotic dominant. If they were recessive alleles, we'd call them homozygotic recessive. All right, so when we talk about homozygotic, all we're saying is on the same locus, on two different homologous chromosomes, okay, the same locus, which is the position, on a pair of homologous chromosomes, if the DNA is the same, the allele is the same, this individual is homozygotic. Now we have another situation here. In this situation, what changed? Okay, so what's changed here? Something is now changed, right? Well, do you notice how the bottom sequence has changed its sequence? All right, what's happened? Well, before we had two of the same sequences, right? They were homozygotic. But now we have two different sequences. Something else has happened. This is what we call heterozygotic or heterozygotic. They have two separate sequences. When we look at the same locus on a pair of homologous chromosomes, okay, they have two separate sequences of DNA when you look at the mother's chromosome and the father's chromosome. Okay, so let's just say we sequenced both of chromosome number 9s from the mother and the father, and we looked at the allele that codes for blood type from the mother's chromosome and from the father's chromosome. And these were different sequences. Heterozygotic. All right, so we'll say that term. We've said that term a lot in class. That's what we're referring to. So let's just say this was an O. Okay, we'll call this the O allele. So we'll say this sequence right here, this sequence down here that begins with G and ends with C, this sequence right here, we're going to give the abbreviation of O2. Right? And we've drawn A's and O's and all sorts of different alleles this semester in the past few weeks. All we're saying is that that letter corresponds to a sequence of bases on the chromosome. And instead of us drawing out all of those bases, doesn't make a lot of sense, we just ascribe that sequence of DNA to a particular letter. Right? This makes it a lot easier for us to draw out and to describe this. And this is a situation of someone being heterozygotic. Okay, they have two different sequences on the same locus when we look at two homologous chromosomes. Okay, so again, know the terms homozygotic, and there's both recessive and dominant, and heterozygotic.